Okay. Good evening, everyone. On behalf of ESIC Sign, I'd like to welcome you all to Decoding Brain Waves, an expert-led webinar series. Today, we are on session 2 of day 1 of the series, and we're really excited to continue the discussion with another fascinating topic. I would like to call upon Arya K to give an introduction to ESIC Sign and who we are.
Good evening, all. The student interest group in neurology, SIGN, of ESIC Medical College. SIGN is a student-led academic initiative formed under the guidance of American Academy of Neurology, AAN, with the aim of cultivating early interest and deeper understanding of neurology among medical students. In ESIC Medical College, SIGN was established with a vision to neurology beyond textbooks. Under the guidance of a faculty advisor, neurology, who provides academic mentorship and direction, the student body is led by a dedicated core committee, ensuring smooth coordination and active student participation through academic discussions, case-based learning, expert lectures, and awareness programs. SIGN encourages students to develop strong clinical reasoning in neurology. Journal Club under the ESIC sign helps bridge the gap between research and clinical neurology while nurturing a strong academic foundation among students. To conclude, SIGN is not just a group but a shared journey of learning and discovery. With passion, teamwork, and mentorship, we hope to make neurology less intimidating and more inspiring for every student. Thank you.
Thank you, Arya. Uh, For our second session today, we have with us Mr. Naveen Keerthi, who is a neuroscience researcher with experience in brain and behavior studies. He is currently a research assistant at GATE Lab at NIMHANS, one of India's leading institutes for mental health and neurosciences. He has also contributed as a neuroscience research assistant at NBRC, that is National Brain Research Center, and has been associated with ACTA. He has worked across different areas in neuroscience, from understanding how the brain functions to studying movement and newer technologies. And with a strong research background, he brings valuable insights into topics like brain waves and consciousness. Today, he'll be speaking about how brain waves can help us understand consciousness, especially in critical states. And with that, I'd like to invite Mr. Naveen to begin today's session.
Are you guys able to see me?
Yeah.
Yeah. So, a very good evening to everybody present here, all of the viewers of this very um, a knowledge-provoking and knowledge feast that Sign has prepared for us here. I am Naveen Keerthi. I'm a final year MBBS student at MIMER Medical College, Pune. And before starting what I prepared for you all today, I would like to thank Mr. Sean Neville and the entire team of ESIC Sign for giving me this wonderful opportunity to share my passion with everybody present here. So just a brief moment, I'll share my PPT with you all. Yeah. Yeah. So, I mean, when we talk about brain waves in general, we a lot of times focus on a lot of clinical aspects of it, like how we diagnose seizures with it, brain tumors, and even to an extent, how the patient behaves in general. But but science is not just about medical science in general. It's not just about how clinics really work or how we diagnose patients, but it's something much more beyond that. So that's why I bring to you the topic, the dying brain, what brain waves reveal about consciousness at the edge of life. I know this topic sounds very vague and more of a certain sci-fi. It has certain sort of sci-fi vibe to it. But once I get into the whole topic and the core of it in general, and what I've been researching upon, and my experience with brain waves and patients who have undergone near-death experiences, and also who have touched death and come back, we will realize together collectively as a group here that brain waves not just reveal a lot about how your brain really works, but a lot of secrets of what your brain holds and how it works when almost all systems in your body are failing. And I know I'm not making much sense right now, but once we're done with the PPT and what I'm speaking, I'm sure a lot of us will be on board with what I'm talking about. So we'll be going over since this topic that when Sean reached out to me and he told me that I should probably present here, I was thinking about what topic should I present, because brain waves is essentially such a complex topic that you I could probably talk hours and hours about it, but we have other very qualified eminent speakers as well who are probably way more experienced than I am, who are talking about how the biophysics of the like the EEG waves and the brain waves and how it really works and how we use it to diagnose conditions. But this topic that I'm talking about is seldom really touched upon. So, and this topic is very complex in nature, and there are many different ways of approaching it. And what I've thought of is a very linear and systematic way of looking at things here. So first we'll be going over the introduction of what brainwaves really are and how they're produced. Then we'll be talking about the methodological approaches to studying the brain, as in what are the current methods that we have to study near-death experience and how the brain really dies in the process of brain cell death and what happens after brain cell death in the body. And then we'll be going over the most important part of the PPT really, which is the EEG patterns of end of life, like when a patient is in the ICU or the patient is going, all the patient's systems in the patient's body are shutting down, how the EEG really works, and how we can use the EEG patterns to essentially diagnose brain death, which is something that's commonly being done, but there's very less research pertaining to it. And then we'll be talking about very interesting translation and theoretical applications of it, future directions that I thought of that would be very interesting to discuss, brain waves and consciousness, which is such an interesting topic, let me tell you. And it's so complex and so beautiful at the same time. And finally, we'll be concluding with what we have discussed so far. So to give you a little background, I'm a final year MBBS student at MIMER Medical College, Pune. But during that, I've done a lot of things. Like I'm the remote research neuroscience assistant at the National Brain Research Center at IISc. I'm a neuroscience research assistant at NBRC. I'm a part of the 3D segmentation team at Neurosurgical Atlas under Dr. Aaron Coyle-Godel, where we basically take 3D scans of the brain and gliomas and tumors, and we reconstruct the tumors to basically create an atlas that neurosurgeons can use during surgery perioperatively. And then I'm a research assistant at Jawaharlal Nehru Research Science Institute under Dr. Anupama Satyamurthy, where I'm leading a gene study where we are looking at the molecular profiles of dimethyltryptamine that's released during near-death experience. And I have almost 20 plus abstract acceptances nationally, internationally in conferences like AN, ANS, and NASS, etc. I just recently completed a psychiatry and neuroscience clinical elective at the University of Kansas Medical Center in the US. And I've been working with ACTREC and Tata Memorial Hospital in Mumbai to essentially uh, progress the research and the background of CAR T cell therapy as well as neuro-oncology. I have certain prizes and I've won certain best research paper awards at international neurosurgery and ophthalmology conferences, recently being ICON 2026. I have done three neuro-ophthalmic and computational modeling science courses at IIT Madras, IIT Bhubaneswar, and I was also a research assistant and research intern at the psycholinguistics neuroscience lab at IIT Bhubaneswar, where we literally were working on how ADHD and eye movements are related to one another. And I'm a research assistant at Sayadri Hospitals. And I'm the youngest review board member of ACTA Scientifica, the Neurology Journal. I'm a peer reviewer of a lot of the journals that I've listed here, the prominent one being NEJ. I have multiple wet lab and data science experiences in the US and India. I'm part of the Aspire cohort at Field Park Medical College, Chennai for budding neurosurgeons. I was selected for the prestigious ICMR STS course and the MUHS STRG. And I'll be going to Harvard this fall. And in the upcoming year, I'll be a microneurosurgery research assistant at the University of Cincinnati, USA. And I think I've spoken a lot about myself now. So let's jump into the topic right away. So even before discussing what brain waves really tell us about death and near-death experience and how the brain dies, I'd like to share a very interesting topic and very interesting case that I saw at the University of Kansas Medical Center that will kind of put all of the this wide topic into perspective on what we're discussing and why we're discussing it in the first place. So when I was working in the University of Kansas Medical Center as an intern, what happened is that one day, one fine morning, we saw a patient who was like 40 years old, and he came with catatonia. Like catatonia is that he like essentially froze while speaking in the kitchen in his house, and he was just entirely stiff. His eyes weren't moving, his limbs weren't moving, and he had come on an emergency basis to the University of Kansas Medical Center. And I was working with the psychiatry and neurosurgery team back then. So what happened is that when I entered to like we take patient history, right? When I entered to take the patient history, what happened is that I noticed something very peculiar. He hadn't spoken for 2 days at a stretch. And once I entered the room, he started speaking continuously. And he started speaking so fast that we could not understand what he was talking about, but we realized that he was in a state of confusion. And then we started to make out what he was trying to say through his thick accent. And he was saying something along the lines of, I want to go back in time. I want to travel back in time. I want to go back in time. Time is nothing but building blocks. And none of this was making sense. Like, does this make sense to you? I'm pretty sure none of you are understanding what's happening right now. And even I wasn't. So later on, within a span of 10 to 20 minutes, we tried giving him benzodiazepines to calm his agitation down, and we had sent his CSF for prion markers. Like a lot of you guys must be aware that prion diseases, like the mad cow disease or Creutzfeldt-Jacobs disease that we call it, like it's one of the rarest diseases in neurology and neuroscience in general. Like prions are just weird things, right? So the prion marker surprisingly came out to be positive, and it was one of the rarest cases in the entirety of the country of US. And we had diagnosed a case of CJD or Creutzfeldt-Jakob disease. And within a span of 10, 20 minutes, the patient passed away, and which was very unfortunate. But there was something very interesting. Right before the patient passed away, we had taken his bedside EEG, and his bedside EEG revealed something very, very peculiar, which normally not seen in the EEG or brainwave patterns of normal people, or even with people who are like terminally ill or malnourished, and etc. And that's what we'll be looking at, and how in the future, probably looking at just the brain scan or the brain waves, you can tell if the patient is going to slip into coma, into cardiac arrest, into shock, or is probably going to slip into a state of a life-threatening condition, and how we'll see. So all of how EEG is basically viewed started with a certain case where an 87-year-old patient developed epilepsy, and Dr. Rahul, who was in the University of Tartu at the time, and his colleagues used continuous EEG, to detect the seizures and treat the patient because the patient was having seizures. And during these recordings, the patient had an heart attack and the patient passed away. But this was very unique because the patient passed away while EEG was being recorded, and this allowed them to see what happens when the patient passes away and the EEG is being recorded at the same time. And they noticed something very peculiar, that through generating oscillations involved in memory retrieval, the brain may be playing a last recall of important life even just before we die, similar to the ones reported in near-death experiences. And I know near-death experience is a very controversial topic. Some of the scientists agree on it. The scientist that I'm working under does not agree on the existence of something called as NDE. I'm a huge fan of it, but because I'm a huge fan of sci-fi in general. But regardless, these findings challenge our understanding of what exactly happens when life ends, and how a brain behaves when it goes into a certain stage of what we call as brain death, and which raises a very important question, that what happens when the brain really dies? Like how do the brain waves really change? And to understand that, we need to look at how the brain waves are generated and what they really are. So please bear with me. I'll be going through certain basic concepts of how the brain waves are generated, but that's important because we are going to be looking at certain very complex topics that for which to understand we need to look at the basics of how brain waves are generated. So before I go into the core discussion, there's another peculiar case, which is famously called as the case of patient one, where there was a 24-year-old pregnant woman with a history of cardiac arrhythmia who suffered cardiac arrest at home, which led to prolonged cerebral hypoxia. And cerebral hypoxia, brain cannot tolerate it at all. The brain goes into certain neuronal death. So she was resuscitated after multiple defibrillation attempts, but she remains deeply comatose with severe cerebral edema and no neurological responsiveness. And after 3 days that they kept her in the NICU, like the neurointensive NIICU, the life support was withdrawn.
And during this period also, her continuous EEG monitoring was done, and it captured the moment of death that happened, the brain death that happened. And it revealed high organized frequency gamma activity and increased neural coherence and withdrawal of after withdrawal of ventilator support, all of which we'll be reviewing later on. So this observation challenged the traditional view that neuroscientists and neurosurgeons and neurologists had that of a purely suppressive dying brain, and suggested that there's a possible opinion of a structured neural activity in the final phase of life, which is very radical at that point of time. So what are brain waves? Brain waves refer to certain electrical activity of the brain. The brain is an electrical organ, right? How the heart is an electrical activity, the brain has an electrical activity as well, because the brain contains trillions and trillions of neurons. They say that the brain has more neurons than the number of stars in the galaxy, and that's very crazy if you look at it. So it records the electrical activity of the brain, which is characterized by different frequencies, which range from zero point 0.1 hertz, which is very less, to about 100 hertz, with specific classes like beta, gamma, alpha, theta, etc., and delta, which is associated with varying states of alertness, cognitive activity, and sleep. And because of the fact that in the brain, which is a very complex organ, which even seasoned neurosurgeons and neurologists do not understand completely, because the brain is such an evolving structure, an evolving organ, and all of them fire simultaneously to make who you are and who I am. They change the electric potential of the brain is actually large enough that you can record it using electrodes which you place on the scalp, which is something very similar to what we do with the heart, which is the ECG, right? So we call this the EEG, or the electroencephalogram, or the electroencephalography, and which records the changing electrical potentials of brain waves that fluctuate between positive and negative values. These waves can be described, for example, in terms of the size. They can be described in terms of the amplitude, in size of the size of origin, site of origin, and also the frequency, which is measured in cycles per second, or what we commonly or very famously call as the hertz. So before getting into how these brain waves are affected, there are five different types of brain waves that we need to ponder upon. One is the gamma, which is associated with high-level information processing learning. For example, when you read Harrison, for example, in medicine, what your brain really does is that it produces a lot of gamma waves. And then there's the beta waves, which occurs when you're talking. Like when I'm talking right now, my brain is continuously generating a lot of beta waves. And if, let's say, I close my eyes right now, I'm closing my eyes right now, and now suddenly my brain is going to start generating alpha waves, which gets secreted when you relax, calm, and when there's no sort of visual or auditory input that's there. So when you open your eyes, when you're talking, there's beta waves, and when you're calm, when you're sleeping, there's alpha waves, and when you're trying to understand something, there are gamma waves which are produced. And when you're very sleepy, let's say you have an exam tomorrow and you're very sleepy and you're trying to sleep and not able to sleep, and like you're very drowsy, and you're studying at 2 a.m. at night, you have an exam the next day, what gets released is something called as the theta waves. And finally, when you dream, when you go to sleep, and when you're like, oh, fine, let me go and sleep. I have an exam tomorrow. And when you're sleeping, what gets released is the delta waves. So every certain phase of the brain, or everything that your body goes through, the brain has a corresponding wave that it produces. And this is very important. And we'll come to why. And there are very several techniques of measuring brain activity and inferring brain waves. One very common, very tried and tested method being the electroencephalography, which records, as I mentioned earlier, which I was talking about, it measures the electrical potentials directly from the scalp, which measures about milli-level or microsecond level temporal resolution. And then the second part is the magnetoencephalography, which is MEG. It is it's an expensive procedure that's done. I saw it being used at the University of Kansas Medical Center when I was there in the US, and I was rotating through a lot of neuroscience labs. There they use it because of the presence of having a lot of funds. Even places like IISc and NIMHANS tend to use it, but again, the use of it is not that popularized because again, it being that complex and very of high cost. And it detects the magnetic fields which is generated by the neural surface. Because whenever current flows, we all of us have learned in our 11th to 12th physics that whenever current flows, it creates an electromagnetic wave around it. So this is what an MEG uses to its own advantage to kind of look at the brain waves. And then there are the non-specific tests like the fMRI, the PET scan that you do for brain scan. Like what the fMRI really is, that it looks at the amount of blood that your brain receives when it's activated. For example, when you're trying to study, your hippocampus is going to receive a lot of blood. I'm sorry. And when you're trying to like sleep, your temporal lobe is going to receive a lot of blood. Like when you're trying to think, when you're trying to draw, when you're trying to be creative, your frontal lobe is going to receive a lot of blood. So these changes in the blood supply through the different parts of the brain is kind of recorded by the fMRI. I have worked with fMRI before, and let me tell you, it's very expensive. And fMRI, it like kind of offers a very detailed analysis of what is going on inside the brain of yours, but it's very expensive to do, but and has very poor temporal resolution. So it's not like it's done usually, but it has other uses like for other functional uses, for uses that talk about how the brain really functions and which part of the brain functions more, which brings us to the next test, which is the PET, which is the positron emission tomography. This is done mostly for, like all of you guys know, it is done mostly for tumors, but because you give essentially a substrate of glucose and you see which part of the brain and which part of the body is taking up the glucose the most, and which essentially shows that, oh, this part of the brain or this part of the body is metabolically more active because it is taking up more glucose, right? But if you look at all of these tests, the one that shines through, the one that we are going to talk about the most right now in this talk, is EEG, or electroencephalography. And it's considered superior because of a variety of reasons. The neural activity, it records neural activity in real time, it is non-invasive, it is inexpensive, it can be done at the bedside of the patient. It offers a lot of data regarding what's happening in the brain, what's going wrong in the brain, what's going right in the brain, and it defines certain brain structures as well. And therefore, is a more practical and a more widely used tool for both clinical purposes, academic purposes, and research purposes involving brain rhythms. So, what is brain death really? Brain death is a permanent, irreversible, and complete loss of brain function, which includes the cessation of involuntary activity, which is necessary to sustain life. All this is the definition that we read in FMD, right? Like forensic medicine, and it differs, and neurology as well. And this differs from persistent vegetative state, in which the person is alive and some sort of autonomic functions and brain functions remain. This is also distinct from coma as well, because in coma, your brain is essentially in a state of resting activity. And because some sort of brain and bodily activity also remains, but it is not the same as locked-in syndrome. Brain death is a medical legal death of a person due to complete and irreversible loss of all brain functions, including the brain stem and the brain stem reflexes, which is how we test if the doll's hair reflex and the calories test, which is how we test if the person is brain dead or not, if the patient is brain dead or not. And it is this definition of brain death is complete, but it is not really accepted in a lot of institutions. Like, there are still debates going on how you should define brain death. There's a black dictionary that provided us different definition of brain death. So brain death is a very hot topic. Like, different doctors have different views on what brain death really is, like how to diagnose it, how to know if a patient is brain dead. There's a lot of ethical and medical considerations surrounding it. It is currently being investigated by numerous institutions and numerous organizations using cellular methods, computational methods, mathematical methods, and animal models, to name a few. Like right now, like next year, I'll be going to a lab in Harvard that is exactly going to be working on just this. And there's also a certain lab that I'm going to visit, which is in the University of Pennsylvania, UPenn, which is working on how the brain looks at the past, present, and future using all of these methods and brain death essentially. So how, like, what is the connection between brain waves and the dying brain? Like, how are they connected to one another? Because this seems like a very peculiar topic, right? Why would some random guy come and talk about the dying brain of all of a sudden? The dying brain and the brain waves, they share a very interesting relationship with one another. The understanding of the brain waves is very essential to understand what happens when the brain dies, because the brain is a very mysterious organ. It is as mysterious as the guy next door, and you don't know what's happening with it. So the brain waves is like a window through which you can peek into the brain and see, oh, this is what's happening. So understanding neural activity at the end of life remains a very fundamental gap in the clinical neuroscience. Like when I was in my first year of MBBS, I had this idea where you could essentially use dimethyltryptamine as a marker to know whether the brain is dying, and how people say that in the last 7 minutes that you're dying, you essentially relive your brain, you relive your life again, like you relive the best moments of your life. All of these, some people say that these are conspiracy theories. Some people say that no, these theories actually exist. So I had this idea in mind. So the way how I took this idea forward was through EEG activity, and which I'll be talking about further. So the neurophysiological correlates of consciousness are extensively studied in states like sleep, anesthesia, coma. Like anesthesia, for example, let's take anesthesia. Everybody, like all of us know that anesthesia is used on a day-to-day basis in all the in all the clinics, all the ICUs, in all the multi-specialty hospitals for surgery. That is basically a part and parcel of how hospitals work, right? But if you go and ask, let's say, an academician of, like, doctor, how do you think the process of anesthesia really works? Like, what is the neuroscience mechanism of it? Nobody has a clear answer. Like, do you understand? Like, we use anesthesia every day, like literally every day, to understand, to treat patients, to diagnose patients, but like, we don't even understand how anesthesia works, right? So there are some studies that say that the way your the brain behaves when it's under anesthesia is how it behaves when it's about to die. So that's probably the reason why we don't understand how anesthesia really works. But it's intriguing, like how we use it every day, and yet we understand so little about its mechanism of action. So But I digress. So, the temporal dynamics of brain activity during the dying process of brain remains very poorly characterized. Like there's emerging evidence which suggests that the presence of transient, organized physiological activity in the which is present in the perimortem period, like about when a patient is about to pass away. Like these observations raised very critical questions regarding the persistence, alteration, and dissolution of conscious processing of the end of life, because because we are still striving to understand what really happens. And one very tempting model, like a tempting way of understanding how all of this works, like what consciousness really is, what you and I really are made of. It's the neurobiological correlates of consciousness that I'll be talking about, which I'll be expanding on more later. Like the neuronal correlates of consciousness are the minimum neuronal mechanisms which are jointly and minimally sufficient for any specific conscious experience. Like for example, for you to have any conscious experience, like like when somebody touches you, when you feel something that's hot or cold, when you're trying to see something, when you're trying to feel something, when like there's a gush of wind that brushes past your hair, all of what you see, like the tactile stimulation, the gustatory stimulation, the audio, visual stimulations, the minimal neuronal mechanism activity that you require in your brain to feel all of these things for your brain to essentially function on a baseline level is what neurobiological correlates of consciousness means. It's a very complex topic. I've tried to dilute it a lot here because it's very difficult to understand even at a level that all of us are, or even for scientists, and even at a level that I am. But it is very important to distinguish a full NCC from substrates which support it in general, irrespective of the specific content. The content specific correlates of consciousness, like touch, like faces, like states of dream or imagination, or like background conditions like consciousness, which contribute directly to the content of experience. And I know a lot of what I'm talking right now is going over a lot of people's heads right now, because it essentially has a very non-linear way of how it flows. But this is how the brain really works, right? Because the brain essentially is the most toughest organ to understand. So we have to kind of like jog our brains a little here and try to understand what's really happening. And the best candidate for full and content specific NCC that I just spoke about is the posterior cerebral cortex. And the posterior cerebral cortex, which again houses your occipital lobe, has certain temporoparietal occipital hot zone. This is called a hot zone because there are certain neurons here that fire continuously to kind of give you a certain perception of how things really work around you, right? It's like how like the heart, like the way the heart pays to beat, you have in cardiac arrest. Like something might happen to you, it's lethal. The same way, if something happens to the neurons in this hot zone, you might stop feeling anything, you might stop seeing anything at that very point of time. It's that important. So there are very two widely used electrophysiological signatures of consciousness. One is the gamma range oscillations and three is the P3B events. Both of this can be recorded using EEG. This is why EEG also speaks a lot about consciousness, a lot about how you feel that you are you and I am me. And we try to see things around us, feel things around us, and essentially observe this universe with our eyes is because of something so little in the posterior cortex called as the neurobiological correlates of consciousness. The very interesting developments, like exciting developments that has happened, where I just spoke about fMRI, right? A lot, what scientists are now working on is that they have combined the whole idea of EEG, brain waves, and fMRI to measure the extent to which neuronal activity is both differentiated and integrated across the cortical sheet, which is very beautiful. It's a very beautiful way of looking at things because EEG kind of tells you how the brain is functioning, at which state the brain is right now. The fMRI tells you about which part of the brain is receiving more blood, which part of the brain is receiving less blood, which talks about the functional capacity of the brain. So you're essentially taking the functional capacity of the brain and the brain waves and merging it with one another to look at the extent to which neural activity is both differentiated and integrated at the same time, to kind of look at how what happens to the brain when it's dying, what happens to the brain when you're under anesthesia, when you're sleeping, when you're awake, when you're talking to people, when you're laughing, and all of these things. So it basically, like, if I have to sum it up in one single sentence, it defines the minimal brain activity that's required for conscious experience. And why is NCC important? Why am I talking about it during when I'm talking about brain waves? It's because the way you see if a person's NCC is intact or not is through EEG or electroencephalogram, which is present when a person essentially when you try to record a person's EEG waves of gamma, theta, and delta waves and see how consciousness works in the person's brain and how connectivity is present in the person's brain. So the dying brain essentially serves as a very natural model to probe the lower boundary of consciousness. Like if you look at consciousness, really, the consciousness seems to have like nobody can. Right now, if you ask me, what do you think is consciousness? I cannot define it. I do not have an answer. I literally do not have a concrete answer to give you that would satisfy you. But but as scientists, our job is to try to look at the boundaries of to try to stretch the boundaries of what we understand and what we know, right? So the dying brain essentially serves as the lower border of consciousness, and trance and meditation serves as the higher border of consciousness. So our consciousness kind of works between a state of meditation and a state of brain death, which is very interesting. So dying brain serves as a very natural probe to probe the lower boundary of consciousness, and till which extent can the consciousness lessen to, and the idea of consciousness lessen to. And why is the end of life brain state so unique? Why did I select this topic? It's because the end of life brain state it represents a rapid, irreversible transition rather than a stable or reversible condition like sleep or anesthesia. Like right now, if you're going to sleep, you're going to wake up 6 or 7 hours later, or early if you have an exam. And when you, if you put a patient on anesthesia, the patient is going to again wake up after some point of time. But why does a person not wake up after death? Like why is that so certain? Why is that a life sentence? Like why can we not revive people after death? Like, why can we not create a Frankenstein? That's because death, even though brain death is very similar to sleep or anesthesia, it's very different. It's very different. Then the reason why we know this is because we tried to record EEG patterns of people who were about to die, and that's how we know that even that initially the EEG patterns of death, like near death experience, or when a person like is about to die, or a patient touches death and comes back, is very similar to the EEG waves of when a person is sleeping, or when a patient is in coma, or when a patient is in anesthesia. It is very different at the same time. But why? It's because it's characterized by global metabolic failure, like the brain is not receiving any glucose at all, which affects all the neuronal systems simultaneously. Like unlike other states, it shows a very dynamic sequence of breakdown, including suppression, burst, and terminal depolarization. And it is often heterogeneous regional variability, with some networks failing earlier than the others. And there's very transient high frequency waves that are present, which paradoxically emerge. Like if you some like if I'm talking about brain waves right now, a certain part of you who is listening to what I'm speaking know what brain waves really are. Like a lot of you guys know the basics of brain waves, and you want to understand more. And a certain sect of you guys do not know what brain waves really are. But if you think about brain waves, even a common man would tell you that if the brain is dying, oh yeah, the brain waves are if the brain is dying, the brain waves are going to decline. But something very interesting happens when the brain dies. Instead of declining, the brain waves start increasing in number. They start increasing in amplitude. It's very weird because the brain is dying, right? The brain is essentially shutting down. But instead of the brain waves declining, why is it increasing? Like why is the brain activity increasing all of a sudden instead of decreasing in brain death? And that's what we'll try to answer to them. There's also a certain problem here because there's a possibility of real-time subjective reporting lacking, which makes interpretation of consciousness and death very challenging, because you cannot every time you cannot just record a person's brain while they're dying, right? Because you cannot predict when a person's about to die, when a patient is about to die. So essentially recording EEG patterns during that time becomes very difficult. So the studies which have been conducted are very few for us to really comment anything, but still it's interesting. So together these features make it a very non-replicable time compressed model of how the brain wide function collapse occurs. Now, I'll be talking about the various methodological approaches that we have developed over a period of, like, let's say 100 years to study the dying brain. Like, they're still very under investigated, but we have certain ways of approaching it. First and foremost, obviously, the reason why I'm here in front of you all is the EEG, or the brain waves. The continuous EEG recordings reveal that the human brain may be capable of generating coordinated activity during transition period to death, even after the suppression of neuronal activity in both hemispheres following cardiac arrest. Following cardiac arrest, which is a form of death, obviously, and during near-death experiences, they noted that there's lack of understanding of the neurophysiological process of the dying human brain. Like, we don't understand what really happens. There's experimental animal studies that have been done about this, which shows that there's an increasing gamma band activity following cardiac arrest. But to date, no study has investigated this in humans, because obviously there are a lot of ethical concerns regarding. You can just go and ask, you just cannot go to a ward and be like, this patient is about to die. Let me record their brain waves. It does not work that way. It's not that easy. There are a lot of ethical concerns that are involved. There are a lot of ethical processes that are involved. There are a lot of ethical permissions that you need to obtain. So this is a certain barrier why a lot of people are very afraid of approaching this topic, because there's a very large extent of an ethical consideration involved. But still, there's subjective descriptions of near-death experiences where, let's say, a patient was going into cardiac arrest and they suddenly got resuscitated by CPR, which includes a very memory recalled paranomic life review. So there's a hypothesis that the brain may generate a memory replay within this unconscious space with an increase in oscillatory activity. I earlier spoke about something, right? There's this theory that we talk about that when we are about to die, what suddenly happens is that in the last 7 minutes, they say, like there's a lot of spiritual texts that say this, even religions like Christianity, Hinduism, Jainism, they all talk about like religions. They talk about the certain concept that before you're about to die, the last 7 minutes that in the last 7 minutes you kind of relive your entire life. Earlier, people used to think that, oh, this sounds like a cool sci-fi idea. This sounds like a cool sci-fi books idea. But now we've actually found scientific evidence that suggests that before you're about to die, you actually have certain memory recalls. So probably that's what really happens, right? We don't know. So let's move forward. There are certain animal models of dying brain, like how experimental models of brain death, which was produced by like increasing the intracranial pressure by inflating an intracranial placed balloon catheter, which are very well established and used in transplant associated studies. Like if you look at it, there's no study that really investigates brain death. Brain death happens as a consequence of what you're investigating, and that's how we understand a lot about what happens when the brain dies. Because if you look at it, neuroscience in general, whatever we know about neuroscience right now, whatever we know about it, we know about it because there were certain experiments that were carried out that shouldn't have been carried out. I know this sounds like a paradox, but think about it. A lot of what we know about the brain right now, we know it because there were very unethical experiments done, right? Like, for example, how do we know that every lobe of the brain functions differently? How do we know that? We know that because there was a railway worker back in 1800s who had a railway accident where this a rod this big entered through his eye and entered out of his head. But the patient was still alive. The patient was still speaking. And after that incident, the patient's personality, where after the rod was removed and the surgery was done, and at the time we didn't know anything about the brain and the lobes of the brain, the patient essentially had a complete change in his personality. He would randomly start saying cuss words. He would randomly start shouting at people. He would randomly get angry. He tried to murder his own wife. So what do we understand based upon that? We then understood that the rod had gone through his frontal cortex. And that's why he started behaving like that, because again his frontal cortex was destroyed. It's so interesting, right? And that's why, because of that thing that happened with him, this gruesome, terrible, unfortunate incident, we understood that frontal cortex and the frontal cortex is involved in your personality. That makes how you are, that defines your likes and dislikes. So a lot of you that are watching this lecture might think that why are we doing this to animals? Like, why are we increasing the ICPs and why are we inserting random balloons inside their heads and killing these animals? We do that because at the end of the day, this is the only manner through which we can actually study brain death. We cannot do this in humans, and no matter how unethical this is in animals, this is done because the consequence of this is that we get to understand a lot about the brain. So coming back to this, the aim was to develop an experimental mouse model of the brain death and to compare the explosive and gradual brain death. Like, pay attention to the word explosive. Induction under ICP monitoring. Brain death can be induced in a pig model by inserting a catheter after the trephination of skull. And according to standard guidelines, the brain death diagnosis was established after a flatline EEG, like the EEG has no activity, after which has occurred in all animals at 60 minutes after induction of the balloon catheter. And finally, we have cellular models of the dying brain. Like we first spoke about the EEG, second we spoke about the animal models, and now we are looking into the microscopic picture of it, like the cellular model. Now, if you look at the cellular models, they're not really satisfactory, right? Because cellular model of brain, that is something that happens in Alzheimer's, it happens in Parkinson's, it happens in frontotemporal dementia, it happens in cases like Niemann Pick, it happens in Pick's disease, it happens in like the young onset Parkinson, it happens in multisystemic atrophy, like you just name it, it happens in Prime disease. So in all of these diseases, if you see, they have very different manifestations, they have very different etiologies. But if you boil it down to the cellular models are essentially all the same. Like this is essentially what you read in, like, I don't know how many of you guys read Robbins in second year, like the med students that I'm talking about. In Robbins, there's a very beautiful chapter about cell death and what happens during apoptosis and necrosis. And that's essentially what happens in brain death, right? Because brain death is essentially the apoptosis of and the necrosis of the brain cells. Like what really happens is that due to oxidative stress, inflammatory cytokines get released, the TNF alpha, FAS, and FAS ligand, which is also called as the death ligand, after intracranial hemorrhage, which activates the intrinsic capsid-dependent pathways to induce the emergence of mitochondrial membrane permeability that releases the causes the release of cytokine C, and all of these are the chemicals which indicate the cell is dying, and the goes into a state of death, right? And the commonly studied model through which we kind of talk about brain death is intracranial hemorrhage, because you see stroke everywhere. Like if you go to your ward tomorrow, you might see at least a patient of stroke there admitted in the neuro ICU, right? That's how common stroke really is. So since stroke is so common, the scientists were like, what if we look at stroke? What if we look at brain bleed and think about brain death? Like, how does that sound? And the other scientists were like, that's interesting. And that's how they came to this idea that probably we could look at intracranial hemorrhage as a model of how the brain really dies. And then think about, oh, probably this is how the brain dies. Like the TNF alpha gets released, fast ligand gets released, the NMDAR receptors get activated, ATP gets produced by mitochondria, N-methyl aspartate receptors get activated, TNF alpha receptors get activated, the BAC, BCL2, BCF, BAX, BCL2 associated proteins, the calcium 2 plus related channels, there are extensive molecular pathways which are involved in apoptosis and necrosis that come into play that cause brain death. Which and why am I talking about all of this? Like, why am I talking about this in such depth? Because what if in the future we have a certain chemical in our hands that can stop this, that can stop a patient from undergoing brain death. And in the future, finally, like 300 years down the line, there might be a time when we can literally stop brain death. We can stop death in a person. Death lives us all. Like in this society, if you see, death is the most certain and the most common thing that all of us see. So what if suddenly one fine day science finds a cure for it and we attain immortality? All of these secrets that you find in the brain, all of these science fiction stories that you read might someday be just literally possible because just because we read more about the cellular mechanisms of how the brain really dies and the EEG mechanism, right? And we are like investigating more on it, and we are finding some very interesting uh, results. So now I'll be talking about what I'm really here for, which is the EEG patterns of end of life. I'll be going over certain literature review that I did of the certain papers that I went through for this topic, and I found very interesting topics that talk about how the EEG really changes when the brain dies. So one really interesting paper that was just released 2 years back, like a year or so back, which was in the neurology education journal, any journal, which I am a peer reviewer of, and I came across this paper, which is basically the EEG of the dying brain, where a retrospective analysis of a severely terminally ill patient undergoing VEEG, or which identified three distinct phases of brain activity during the dying process. Like what they did is that when the brain was dying, they luckily managed to record the EEG activity, and they noticed that it does not just happen randomly. Like it's not like random things happen when the brain dies. Not random things don't happen. Something happens in a very structured, very systematic manner, and it can be divided into three phases. Phase one showed there was theta delta activity. Phase two showed burst suppression patterns, and phase three was characterized by terminal background suppression until death. So basically, what happens is that in phase one, there's theta and delta waves predominate, and in phase two, which is the main phase, gamma waves predominate, like the burst of gamma waves, and in phase three, the brain dies. The EEG comes to a flat line, right? So these findings suggested that there's a progressive structured electrophysiological decline rather than paradoxical activation of the dying brain. Like initially, people thought that the brain just dies, like the EEG just disappears. And then they investigated and found out, no, the brain does not really die. Like there's paradoxical activity. And now, recently, this is the latest evidence that suggests that no, that's also not true. The brain goes into phases of death. And during these phases of death, there are certain changes that are noticed with every phase. There's another paper that was released way back in 2011, where they tried to essentially look at EEG patterns in patients who have near-death experiences. But I'm talking so much about near-death experience, near-death experience. What is a near-death experience, right? A near-death experience is a certain mechanism of like a certain phenomena where your body almost touches death and comes back, right? Like your body almost shuts down and comes back. For example, let me give you an example. There's a patient of sepsis that you're treating aggressively with antibiotics, and the other doctors are like, oh, this patient's survival does not seem that good, right? There's like 20% that this patient will survive, and he ends up surviving. Let's say he ends up surviving. The patient has had a near-death experience. Or the second example being, let's say there's this guy who's like driving his car, Like he's driving his car at a very fast pace and he ends up crashing. He ends up fucking crashing. And what happens is that when he crashes, the the his brain goes into a certain death, like a certain frenetic death. And when he frenetically crashes, the brain waves, what happens is that these brain waves get recorded, and when you look at the brain waves, you see that the patient is going to die. But no, the patient has not died. The patient has gone through something that is called as a near-death experience, right? So, like we see and hear about a lot of these near-death experiences. And there's this doctor in called Lakmir who will, along with the studies, try to report the patients who were at their end of life, who were on intubated support, and you withdrew their life support without any medications, without any IV infusions, without any machine ventilation. They exhibited a surge of EEG activity that I just spoke about, like the phase 1, phase 2, phase 3. These guys did not know phase 1, phase 2, phase 3 because that the research that they're doing right now, the paper that I'm talking about, is essentially 13 years before when we know about these phases. Like we came to know about these phases in 2024, and this research talks about something that happened in 2011. So in 2011, they did not have any concept of phase 1, phase 2, phase 3. All they knew was that, oh yeah, the brain, what happens there when the brain dies? There's a certain activity, spurge of activity that is seen in the brain waves rather than the decline. So the researchers at that time speculated that there's a very similar surge of electrical brain activity that is seen in the patient of brain death also. So they're like, okay, probably near that experience, something happens to the brain that is very similar to brain death. It's not the exact same as brain death, but something that's similar to brain death. And they noticed that the observed EEG surge appears to be sufficient duration and strength as to account for the vivid experience that is seen in ND. So people, like, there are very different accounts of people undergoing near death experience. Like if you talk to them, if you read articles, if you think, like, if you go through the review of literature, you see that a lot of these patients who have near death experiences, they report something that they say that, oh, I saw God, I saw tunnel, I saw light, I saw certain beings who came to me, I saw my parents, I saw my children. How do they see all of these things? Are they lying? Are they lying to you? No, they're not. Why would they lie to you? Which means that probably they are seeing those things, and they are seeing those things. And how do I know this? How do we know this? We know this because we are able to see excited brain waves, excited gamma patterns in these patients, which shows that, oh, probably they are seeing these things. Their visual cortex is getting activated. They are hallucinating, probably. They are seeing things that is probably not real, but they are seeing it for sure. Which makes things like the 7-minute death experience or near death experience or seeing God or spirituality even more real, right? So there's another paper that was produced in 2015, which was a recent report of electroencephalogram of a dying human brain, which was in 2022, which reported a transient increase in the gamma power, which coincides with the loss of bilateral hemispheric activity, followed by decline of cardiac arrest. Importantly, there's no sustained rise in absolute gamma activity, which was observed post cardiac arrest. What this essentially, all of what I just spoke about, means that when they were investigating, they found that in the phase 2, the gamma activity was increased considerably, which was very surprising to them because they knew that brain waves were increasing, brain spurges were increasing, there was spurts of activity, but they didn't know that it was gamma. They thought it might be some other wave, it might be beta, it might be theta, it might be delta, but nobody was thinking about gamma waves being the reason being the reason behind all of these wave progressions that happen in the EEG. So what they found out was very radical for that time. Like in 2022, that's when we finally noticed for the first time that, oh, this is also that's something that happens. This is very new to scientists at the time. So this finding challenged the hypothesis uh, that enhanced gamma oscillations underlie phenomena such as life review, suggesting instead of brief state-dependent fluctuation followed by suppression. What this means is that people used to earlier think that, oh, there's brainwave excitation followed by brain death. But what they hypothesize is that no, that's not what happens. What literally happens is that there's gamma oscillations which underline phenomena such as life review, which suggests that instead of a brief state-dependent fluctuation of followed by suppression, there's a stage systematic activity. There's a very interesting paper that I wanted to talk to you guys about, where it's titled "Do Not Go Gently Into That Good Night: The Dying Brain Analysts Paradoxically Heightened Electrical Activity," where it suggests that these signals are footprints of consciousness that would be increased if such large gamma surges in the posterior hot zone could be observed in neurotypical subjects. For example, during unusual experience state that share some characteristics with near-death experiences, like high dose of psychedelics, which can lead to ego dissolution and mystical experience. Like for example, like a simple way of saying this is that A lot of us talk about drug addiction, marijuana addiction, psychedelic addiction, right? Like mushrooms and all of that. Uh, like why do people get addicted to it? Why is there a lot of addiction to marijuana? Like why is there a lot of addiction to drugs like methamphetamines? That's because when you take drugs like cannabis and methamphetamine, your brain goes through hallucinations and illusions and visual and auditory hallucinations that is very similar to what happens when people say that they see God, when they see that they are about to die, when they see that they have a near-death experience. All of these are very similar to one another. So they believe, like scientists believe, that when you take these drugs, something happens in your brain, like the EEG activity of your brain. It's very similar to what happens when people have a near-death experience or when people actually die. So a more direct way to detect covert consciousness in behaviorally unresponsive neurological patient probes the cortex with magnetic pulses and listens to the echo using high density EEG, which I spoke about MEG, right? Magnetic encephaloelectrogram. That's what they did, and they found out that the not just the electrical activity of the brain, the the magnetic activity of the brain also changes. But through all of what we discussed, this one very important wave that is very has been very strongly correlated with brain death and the dying brain and near-death experiences and spiritual experiences and people seeing God, and that's the gamma wave. When the analysis of 4 dying patients in a research showed that there was global hypoxia of the brain. Like what they did essentially, there were certain patients on ventilatory support, right? They withdrew their ventilatory support, and these patients went into brain death. So when they went into brain death, they did an fMRI, EEG, complete thing, and they found out that there was global hypoxia in the brain. The brain was not receiving any blood, any nutrition, any oxygen. And in spite of not receiving any of these things, there was no oxygen in the brain, no glucose in the brain, no nutrition in the brain. Still, the brain was producing transiently high gamma waves, like excessive bursts of gamma waves, which was being produced. And these surges involved both the local connectivity in the temporoparietal occipital regions, like the temporoparietal occipital regions, and also longer range coherence with the prefrontal regions. Like what this essentially means is that the gamma waves were not all localized. They were connecting your temporal lobes, your parietal lobes, your occipital lobes, and then they were shooting to your prefrontal cortex. So they were connecting areas of your personality, your memory, your hippocampus, your vision, your auditory functions, your gustatory functions. All of it was connected to one another. So the gamma wave is not localized as it was taught earlier. The gamma waves in EEG, which are of a certain frequency, involve all the areas of your brain, which involves a long-range coherence of the prefrontal cortex and patterns associated with integrative brain function. And although the underlying mechanisms and significance remains quite unclear, these findings indicate that the dying brain may exhibit organized active network dynamics, where we used to think that something haphazard is happening in the brain. Oh yeah, the theta waves get released and the beta waves get released. But this paper also showed you, this was released in 2023, because they say that in 2022, 2023, and 2024, a lot of research was happening, still is happening. So lot of these papers are talking about like very recent papers, which shows that the brain undergoes something that's very organized, which is very active network dynamics, challenging the notion of immediate neuronal silence after cardiac arrest. So gamma oscillations play a very important role in the dying process of the brain. And we are trying to investigate like where is gamma wave getting produced from? Is it getting produced from the temporal lobe, from the occipital lobe, from the parietal lobe, from the frontal lobe, from the brainstem? We don't know. We don't know. We're still investigating. So still, Dr. Vincent reported paper that I just spoke about, again spoke about a certain thing about gamma waves, where they said that the patient displayed a very temporary increase in gamma power when bilateral hemispheric activity ceased. Like for example, when the bilateral, both sides of the patient's brain stopped working, instead of the gamma waves dying, the gamma waves started increasing in nature. Like, why does that happen? It happened because there was an increase in absolute gamma activity, which is speculated to be responsible for at least life review, but rather showed a reduction of absolute gamma waves after cardiac arrest. So it was only the relative, but there's certain limitation also, right? It's not all. There's nothing there's nothing concrete in medicine. Like some people, like some researchers said that there's only relative amount of gamma that was increasing compared to alpha beta. Like gamma was not transiently increasing. It was not. I mean, it was not increasing in amplitude. It was just increasing when you compare it to waves like alpha, beta, and theta. And all of the brain activity falls after incidents like cardiac arrest because the brain waves like alpha, beta, and theta decreased faster than gamma, and the percent of residual activity was greater compared to the later frequencies. What they try to essentially say is that they're saying that the gamma waves are not probably increasing. It is it's just that the gamma wave is decreasing lesser compared to the other waves. instead of the gamma. So it appears as if the gamma wave is increasing, but in reality, they think that it's just the gamma waves are falling, the falling lesser compared to the other waves, so the other waves are falling faster compared to the gamma waves. So they think that the gamma waves don't increase, it's just that the other waves decrease faster, and it's just a relative aspect that we're looking at. So regarding the source of gamma oscillation, like where do these gamma waves get produced from? They think that the gamma waves are produced from a certain conscious perception, like for the visual cortex, occipital cortex, right? And also from variety of brain circumstances ranging from ongoing brain for preparation and also for execution of certain movements. So they think that the brain waves, like the gamma waves especially, are produced from the occipital lobes, but some researchers say that no, these brains are also produced from the frontal cortex and also the brainstem. So right now we aren't really sure where the gamma waves are produced from, but most people say that it's most researchers try to agree on the fact that it's mostly produced from the frontal cortex and the occipital cortex. So I think I do have some time left. So I'll be talking about the translational and theoretical implications of the brain, where why are we discussing all of this? Like, what does it hold? Like, what does all of this mean essentially? We spoke about EEG patterns. What is brain death? What are the mechanisms of brain death? How do we investigate brain death? How do we use EEG? What is seen in the EEG? But why is all of this important? Like, what does all of this mean? Why does it matter to a clinician? Why does it matter to a researcher? Because number one, it helps in redefining the temporal boundary of consciousness. The traditional frameworks assume that there's a relatively abrupt loss of consciousness following brain death. However, Perimortem EEG data suggests that there's a graded and dynamic transition. There's no shutdown. It's not like switching off a certain. It's not like switching off your TV. It's not like the brainwave randomly goes shut. There's this very synchronized dynamic transition that happens from brain living to brain death. And observations of transient gamma activity, preserved coherence, and network level interactions raise the possibility that there are certain neuronal substrates of consciousness which may persistently briefly persist beyond clinical threshold, which simply in simple language means that you might think that this patient's brain dead, but the patient's consciousness is still living for a certain brief period of time. It's so interesting. So what is death really? Because we say, what is death? We say that death is the irreversible cessation of the functions of the brain along with the brainstem and the respiratory and the cardiac functions. But here, we're knowing that in spite of brain death, the consciousness still persists. So is the patient really dead? Or is the patient alive even after brain death? That's an important question. So this shifts the question from binary state, like dead, alive, conscious, unconscious, to a continuum model of fading neural integration. Like it's not an on and off or a black and white phenomenon. It's more of a gray spectrum that we see when you talk about brain death. That's what all of this means. Second, implications for brain death criteria, like just what I spoke about. You cannot just say that this person is brain dead just by looking at the EEG, right? There are a lot of irreversible sessions of brain functions, clinical examination, ancillary testing of the brainstem reflexes. That's how we come to like there are multiple doctors that are involved, and they come to a conclusion that oh, this patient might be brain dead. But what if brain death is still not has still not occurred? What if the brain is dying? What if there's a certain way to save the brain? What if there's a certain way to save the brain from irreversible cessation of function? So it suggests that perimortem studies suggest that residual electrophysiological activity may transiently rise even after circulatory arrest. Like the patient is dead, okay? Like you're saying that there's an ECG flatline. There's an ECG flatline that we're seeing on the monitor. But still the brain functions persisting. How? How? We don't know how. Like there's EEG activity that's persisting. How? We have no answer for that. But all we know is that in spite of us doctors or medical students, we noticing that the ECG is like transiently low or there's a flat line, the EEG still persists. So even though this activity may not imply meaningful consciousness, we cannot just say that, oh, no, because we're seeing gamma waves, the patient is alive. The patient is obviously brain dead. It raises very important questions in neuroscience and neurology. What does it raise? It raises a question about the temporal resolution of current criteria. Like, is the current criteria of brain death even relevant in the current scenario? Like, with the current understanding that we have of how the brain dies, is it really relevant? Is this really correct? Like, whether early post-arrest activity represents non-functional noise versus residual organization. And importantly, these findings do not invalidate current brain protocols. I'm not saying that the current brain death protocols that we're following in ICUs and medical colleges is wrong. I'm not saying that. But what I'm saying is that there's a need for clearer differentiation between electrical activity and functional consciousness. Like the electrical activity of the brain and the functional activity of the brain is something that is entirely different. And also there's a potential refinement of understanding the sequence of brain failure. So there are insights into, and finally, we also get to see the insights of models of brain consciousness. Like we get to see how the brain consciousness really works, like the global workspace theory, integrated information theory, the thalamocortical loop models. So the brain, the process of brain death and the association of brain waves with it actually helps us understand about how brain death really occurs and what it tells us about how the brain functions normally and what happens when the functions go haywire. Then what's the future? Like what's the future in this topic? Like what is the research happening? What is going to happen in the future? First, multimodal monitoring approaches. Like the future research, I feel personally, the future research must move beyond isolated EEG. Like we shouldn't just look at the EEG. We should look at other things that supplement the functions of brain waves, like fMRI, intracranial microelectrode recordings, for cellular level monitoring. With this combined approach, we can help to distinguish with true cortical signals from artifacts. There's a lot of artifacts that you see. Like when you like, I worked in a lab that was working on EEG, like fMRI and tactography. You see a picture behind me, right? Like a beautiful picture of the brain. That's essentially the tactography that's done. You essentially try to record the MRI of the brain and you do tractography and you see certain tracts of the brain. So, as beautiful as this picture looks, if you think about it, this is not usually done because it's very expensive and it is done by only researchers that are studying brain networks. But I feel this should be done with patients as well. EEG, fMRI, this tractography, all of this should be combined, which helps to map the network level breakdown versus transient integration during the dying process. And critical, which is very critical for identifying whether observed activity reflects functional organization of physiological noise. Second, the real-time ICU data collection frameworks. Currently, data is very largely relies on retrospective and opportunistic data, which limits interpretation. There's need for standardized and prospective protocols and continuous EEG monitoring and physiological monitoring during end of life care. And these precise time locking events, which are also present with development of ethical as well as larger data sets, will allow reproducibility of findings, identification of patient specific versus universal pattern. And finally, integration with computational models of consciousness will also tell us that testing of these theories that I spoke about becomes very easy when you understand that how the brain really dies. And finally, I understand that I've gone a little overboard on time, but I'll just talk briefly about consciousness. Consciousness essentially is conceptualized along at least two dimensions, the level of arousal state and content. Like consciousness is such a complex topic that I cannot just talk about it under 5 or 10 minutes. We would just require an entire session to talk about what consciousness really means. But what we understand right now about consciousness is that consciousness has a lot of certain activities, certain baseline levels, which can also be recorded using EEG. There's putative network correlates of consciousness that I spoke about, the NCC, which kind of tells us about if the brain death has really happened or not. And while it does not really explain the phenomenon, the mechanism and relationship of consciousness experience remains uncertain, right? So finally, concluding, the dying brain is not uniformly silent. It undergoes a structured dynamic transition. Evidence shows that both progressive and suppression and transient surges of organized brain activity occurs. Findings are heterogeneous, they're conflicting. Some say that gamma wave activity occurs, sometimes there's no surge, organized activity is present, global suppression is present. Near experiences finally highlight fundamental dissociation between the level and the content of consciousness. And finally, the boundary of consciousness, I would like to again emphasize that it's not precise. And neural activity of near death may retain short-lived integrative properties. And finally, I'd like to finish with this quote of a very famous neuroscientist that your brain and every brain is a work of progress, right? It's plastic. And from the day that we born, the day that we die, it continuously revises itself and remodels itself. And it is improving or slowly declining, and it depends upon how we use it. So that's it. I'm done with my talk, and I would thank Sean Neville and the eSign team for allowing me to talk here. And yeah, thank you so much. And I apologize for going a little overboard. Thank you.
Thank you so much, Naveen sir, for that insightful and thought-provoking session. I'm sure that we all had a lot to take away from this. I think we can now open the floor for a few questions from the audience. You can just unmute your mic and ask your question.
Hello, good evening, sir. Can you hear me?
Yeah. Hi.
Yeah, so first of all, sir, thank you for the experience. Like the presentation, everything was like personalized and creative beyond anything that I've ever like experienced. So thank you a
Thank
lot for
you.
this. And
Thank you.
second of all, I just wanted to ask, like I was very like in awe when we talked about like your introduction and everything. So I just wanted to
Mhm.
ask, like, what practical lessons like have you learned, like from being like a medical student to getting involved in research that is actually impactful? Because like I'm in first year right now, I'm a medical
Yeah,
student in Jammu.
yeah.
So like
Okay.
it's hard to get into impactful research like initially. But so like what's like the lessons that you might have learned from your experiences?
Yeah, Aryan, right? Yeah, Aryan, thank you for your question. Basically, while I understand that we as MBBS students, when we get into MBBS, when I was in my first year of MBBS, started with an ICMR STS project. And what happened is that a lot of times we feel that as MBBS students or as students in general, we are not able to contribute to meaningful research, right? The research that we do is basically data collection, data analysis, a lot of boring topics in physiology or anatomy. But you need to understand that if you want to do anything impactful, anything in your career that's impactful, you have to start with the basics, right? Like when you start with the basics and when you do the nitty gritty work, like when you do data collection, something as boring as collecting data from patients, you tend to realize that there's a lot to learn from it. And once you're better at it, and once you realize that, oh, I know this well, you can further expand your horizon. For example, I started with an ICMR STS project, which won the grant of around 50,000 rupees. And then in my second and third year, I did a lot of the research methodology courses. I tried to expand my horizon. I tried to break outside my bubble, and I tried to go to other colleges and did my own research. And I tried to understand that finally, I'll just sum it up very quickly. What matters is your lateral thinking. Everybody is going to run behind a certain thing, run in a certain direction. You have to think laterally, right? Medicine is not just medicine. Medicine is physics, medicine is chemistry, medicine is biology, and everything associated with it, right? So try to develop an habit of lateral thinking. Try to think beyond what's taught to you, outside the box, and that's how you will be try. That's how you reach at a question that's impactful and something that you'll be able to do that will benefit the society at large.
Thank you so much, sir.
Yeah, anytime. Any other questions that anybody else might have?
Uh, Excuse me, sir, I had a question.
Yeah.
As you said that the gamma waves are increased while the brain is dying, how clinically
Yeah.
relevant is this finding?
Okay. So basically, when you talk about gamma waves in general and how is it related to whatever I spoke about, is that nothing in science is concrete, like I just said. I might do a research, I might find that the gamma waves are increasing, but another researcher might do it in other part of the world and they might notice that, oh, the gamma waves are not increasing. Does that mean that I'm right? Does that mean that I'm wrong? Or the other person is right? No. It's just that if you talk about clinical relevance, is that in the future, just by looking at the gamma waves of an EEG person, we might try to predetermine when the patient is about to die, right? So we can bring in certain changes where we can salvage the person's life, or we can extend the person's life, or we can introduce in certain therapeutic means or diagnostic means to know when the what is about to happen to the patient's brain or the patient's CNS system. So the potential is immense. The clinical potential is immense. I understand right now it just remains in the area of academy and research. It has not been applied to clinical sciences, but I'm sure in the future, clinicians will be using it left and right to see what's happening in the ICU and when the patient's about to slip into coma and how we can avoid the patient from slipping into coma. The few possibilities are just countless. So yeah, I hope that
But
answered your question.
logically seeing, there might be only fraction of second when there is increase in gamma rays and there is the third phase of brain death. According
Um,
to what you said.
Yeah, no, no. What I said was quite different from what you're interpreting. What you're interpreting is that it just happens for a fraction of a second. It does not happen for a fraction of a second. You record an EEG for a very long time. There are people who record, there are doctors who record EEG for an hour or so. So it does not happen for a fraction of a second. Around happens for 5 to 10 minutes, if you say. It's transient because it happens in spurts, but it happens in a wavelength. It happens about for 5 to 10 minutes or even 15 minutes or 20 minutes. So it gives you a certain time frame to kind of know really what's happening. I'm not saying every gamma wave purge is indicating near death or every near death experience is associated with gamma wave. I'm not saying the specificity or sensitivity is very high, but I'm saying it has been noticed. So we have to do further research. So we do not have many answers right now, but in the future we will. Is what I think.
Thank you, sir. It
Yeah,
resolved the doubt, really. I
yeah,
didn't
yeah.
knew that
Thank you.
there was a time frame between. Yeah.
Yeah, yeah, yeah.
Is there anybody else? I think that is all.
Yeah.
Okay. To conclude the session, I'd like to invite Mayuka to deliver the vote of thanks.
Good evening, everyone. On behalf of the ESIC SIGN, I would like to extend my heartfelt gratitude to our esteemed speaker for such an insightful and thought-provoking session on what brainwaves reveals about the consciousness at the edge of the life. I sincerely thank our moderator for guiding the session so smoothly and ensuring an engaging discussion. I also express my appreciation to all the participants for your active involvement and thoughtful questions, which truly enriched the section. A special thanks to the organizing committee and volunteers for their efforts in making this session a success. With that, we came to the end of the session. Thank you all once again.
Thank you so much. Thank you, sir. I'm sure all of us learned a lot from the session.
Thank you.
We hope to engage with you more in the future.
Yeah, thank you so much for this opportunity. Yeah, thank you.
Thank you.